Become a sponsor today by visiting patreon.com/psychologyis.
But to give a simplistic analogy that people often give, we now have the ability to essentially edit the human genome. And the analogy that people often give is that you can have a Word document and with many words written onto it, many sentences, many paragraphs. And CRISPR is essentially the equivalent of being able to go in, place your cursor wherever you like, delete words, edit the spelling of words, or potentially even add words into it. And again, there are very specific mechanisms by which this works. But yeah, essentially we have, a protein called Cas9 that can be programmed, which can cut and edit DNA sequences. So, again, I know it's not easy because you don't know how far in depth to go with an explanation, but is there anything else on the level of a simplistic explanation that you'd like to add?
I think for the lay person or the the person who's not an expert in molecular biology, the way to think about it is that each of us has a genome. There's information in that genome. It turns out there's about 3 billion individual base pairs, as I mentioned before. And what makes two people different from each other is typically we differ at maybe 1 per 1,000 base pairs. So only a few million out of those 3 billion locations would say Nick, you and I differ at. So there are specific places where our DNA is different, and then there are huge chunks where it's basically the same. And one could just imagine that these wizards in the lab have developed a tool that they can inject into a cell of your body. It goes into the nucleus of the cell where the DNA is and edits the DNA, and it makes the changes, whatever changes are desired. You can design the molecule to make specific cuts or changes to an individual's DNA that are desired. And of course, there's much more detail and subtlety there. There's an error rate. Sometimes the thing edits the wrong site. Sometimes it doesn't work right. But they're improving it. Year by year, it gets better and better, more accurate, more effective. And so I think it's not a crazy at all extrapolation to say that we'll soon have extremely accurate, extremely safe gene editing technology. It is already being used today to cure diseases like sickle cell anemia, et cetera. We can get more into that, and it's certainly in the documentary. I also want to recommend the documentary. Even though I don't particularly like the way they portrayed me in the documentary, I do think it's an excellent documentary. And at the beginning, when they explain the sort of molecular dynamics of how CRISPR works, it's extremely well done, and the graphics are beautiful, and everything is beautiful about the way they explain that. So as a kind of scientific educational introduction to CRISPR, I agree with you. There's nothing better than the first hour of Human Nature.
So many directions we can go with this, but one thing that I think is worth saying here in the beginning too is there's kind of two topics here that we're addressing. One is genetic testing, advanced genetic testing, and then the other one is genetic editing, and those are very different. I think those sit differently with people, because genetic testing is simply gathering information, and I don't know that anyone would make an argument that there's anything unethical about genetic testing. Perhaps you've heard arguments though, but I was very intrigued by something that I understand you do at Genomic Prediction, which is, you have two parents who want to conceive a child, and you help them conceive by basically doing in vitro fertilization with gametes from each of them, and then you'll basically create up to 10 or so viable embryos, which then you can, and of course, as people may or may not know, each embryo will have different DNA. They're pulling different DNA chromosomes from each parent. So each of those 10 embryos, although they're from the same two parents, will have a different genetic profile, which you can see in pretty significant detail. So then the parents can technically choose which embryo to implant. Is that correct?
Yes. Do you mind if I just give a short intro to your
Please,
audience on this?
please.
So there are two aspects of this. One is the whole area of in vitro fertilization, which predates our company. Our company is a young technology startup, whereas the oldest person who was born through in vitro fertilization is now in her 40s.
Mm.
So that's an older technology. It's been around a long time. Currently, in some countries, like for example, Denmark, about 10% of all babies in Denmark today are born through IVF.
Mm.
In most developed countries, the typical number would be something like 5%, 3 to 5% maybe. And so that's a substantial number of kids. That means if you go to a playground, if you go to a kindergarten or a schoolyard, there will be multiples of IVF kids running around. So there are many families that wouldn't have a kid if it weren't for IVF. And it's typically applied in cases where there's some fertility issue. Typically, a very common situation would be the mother is a little bit older, and women's fertility tends to fall off actually much earlier than people think. It actually starts falling off in their 30s. And so already a woman who gets married and has a career and at 35 is ready to have her first kid, it would be very common for that woman to, for example, need IVF. Mm-hmm. So that's a very widespread technology. Obviously, 10% of the babies in some countries are being born through IVF. It's widespread. People are using it all around the world. And I think by and large, everyone agrees it's a super positive development to be able to do this. Many people have benefited from, many people exist today who wouldn't have existed otherwise. But when IVF was first developed, it was extremely controversial. And the original scientists who developed the technique, and a couple of them went on to win the Nobel Prize in medicine for it, they were reviled as monsters at the beginning. So they called these kids test tube babies. I'm older than you probably. So I remember watching as a kid on television. I think I was having breakfast with my family as a kid, and on TV, on CBS News, they were talking about the first test tube baby, and it was a huge deal. It was a sensation. So kind of similar to like CRISPR now or something like that now, but it's become normalized. And the lesson I'm trying to draw from that is that it could be a radical technology that, wow, this baby was conceived in a test tube, artificially outside of the mother. And yet it's become normalized. It's become so normalized that we all have friends who maybe we don't know it, but they had their kids through IVF.